wondered if you'd be like. Hi. Oh, okay, good. I'll catch you. Oh, that's sweet of you to come to this.
My wife had to run out of town this week.
Oh, so you're running around with
so I'm
kids?
a single parent.
Oh my goodness.
So I got a sitter. So it was close.
You could have just come to dinner. You know this stuff.
It's all or nothing.
Evening. I'm Jerry Rubin. I'm the director of the Janelia Research Campus. And on behalf of Howard Hughes, I'd like to welcome you to tonight's Dialogues and Discoveries lecture. I want to begin by a brief safety announcement. I understand we're under a tornado watch, and it's possible that during this next hour, we'll go to a tornado warning. If that happens, you should be very pleased to know you are in one of our official shelter-in-place locations in this building. You're surrounded by 2-foot concrete walls, and this is where we'd send you if you were in the building and there was a tornado warning. So if the alarm goes off, you should not go anywhere. And it may just make a bit of noise during the talk, and we'll shut it off, and then we'll carry on. Okay, so no panic, please. So today's speaker is Bonnie Bassler. Bonnie is a professor at Princeton in molecular biology. She's also chair of that department and a Howard Hughes medical investigator. You should have picked up one of these on your way in, which tells you all the honors and awards and accomplishments that Bonnie, or some of them, has accomplished during her career, and I won't repeat them to you. She not in this report was the fact that she grew up in California, about 20 miles inland from San Francisco, which is why she told me she went to UC Davis for her undergraduate degree, because it was just far enough away from home that her parents, she wouldn't have to live at home and her parents would have to call first before visiting, but close enough so she wouldn't be scared. So I think you're going to see when you listen to Bonnie's talk, she isn't really scared of anything today. Okay, Bonnie, I'm not going to say any more than that because I don't want to use up your time. And your title is Tiny Conspiracies, Cell-to-Cell Communication in Bacteria.
Oh. Okay, thanks. Well, thanks, Jerry, for the interesting introduction and actually for the generous invitation. I'm delighted to be back and to get a chance to give this talk. And I thank all of you guys. You are intrepid for coming out in this weather. So I hope I can entertain you and also teach you something and make it worth your while. So thank you for having me. So I only have one goal for the whole talk, and that's to try to convince you that bacteria can talk to each other. And if I manage to meet that goal, some secondary goals are to try to convince you that the bacteria are multilingual. And then the third thing I'd love you to think when you leave tonight is that actually learning about how bacteria communicate with each other is important, because maybe we can make them talk when we want or not talk when we don't want them talking. And those could be new kinds of therapies or new kinds of products for industry, so that it's actually sort of practical to study how bacteria talk to each other. So that's where we're going. And I thought what I would start is to make sure that we all understand about the critter that I love the most, which are the bacteria. And so my first slide is just a cartoon of a typical bacterium, like an E. coli bacterium that might be living in your gut. And all bacteria pretty much look like this guy. They are single cells, but they're so small you need a microscope to see them, right? And they're all covered by a membrane, which you can think of as their skin, that keeps the outside world out and the inside world in. And inside, in this cytoplasm, is sloshing around all of the biomolecules that make the bacteria alive. And bacteria are the oldest, most primitive organism on the earth. And what's special about them is that they don't have a nucleus, and they have just one piece of DNA, a chromosome, in this cytoplasm. And on this DNA, this blue stuff, that has all of the genes, or the bits of information, that give the bacteria its physical feature, but also all of its behaviors, similar to how our DNA encode those things for us. And so bacteria look very primitive. Their genome is one chromosome. They have a couple thousand genes, so they don't have very many bits of information. And they've been studied for 400 years. And because they're so primitive seeming, for 380 of those years, people thought that they lived as asocial recluses. And what I mean by that is when they divide in half, so the way bacteria grow is they simply double their size, they divide themselves in half, and one cell becomes two, two become four, and so on and so on. And so it was always thought that because they're so primitive, when they divide in half, each sister cell has no knowledge, if you will, of its sibling. So they simply act as individuals. But of course, bacteria are enormously powerful on Earth. And so I want to try to convince you that they couldn't accomplish all of the tasks that we know they do if they simply acted as individuals. And so then before I get to that, I want to try to convince you that you should care about bacteria. And so in order to do that, I want to tell you a little bit of something about your relationship with bacteria. And this will give you this sort of Bonnie Bassler-centric view of the world. So what I have here, this is supposed to be a human being. And what I've done is made all these circles that are supposed to represent some number of your human cells. So there's a few trillion cells that make up a human being. So that's how many cells you need to make you and let your body do all the things that it does. So it turns out that a few months after you're born, at any moment in your life, you have 10 times more bacterial cells than human cells in you or on you. So if you want to use this metric, at best, you're 10% human. You're actually 90% bacterial. But of course, it's the 21st century, and so it's not all about the cells. It's about this magic molecule, the DNA, that has all of this information that gives us our blueprints. So I've remade my man with the A's, T's, G's, and C's of this famous double helix code. And so, of course, the human genome is sequenced. So we know there's about 25,000 genes in a human being. So that's how many bits of information it takes to make a human being. So it turns out that you actually have 100 times more bacterial genes than human genes. So you can maybe use this metric to think about yourself. This makes you 1% human. I think this is actually more realistic. You're 10% human by cells and you're 1% human by genes. And all of these bacteria that have all of these genes that are living in you and on you, they are not just passive riders. It turns out that those 25,000 genes, the human genes, are not enough to keep you alive. These bacteria supply all kinds of products that your own genome doesn't encode, and we need these products to keep us alive. So what we now get, and this is in the past couple decades or so, is that there would be no life on Earth without these bacteria. So no plant, no animal, no insect, no human, because all of us live in this consortia with these bacteria, and they're sort of our 24/7 partners helping us to stay alive by making things that our bodies need. So that's to convince you that you should be thinking about these bacteria. And then also what I want to convince you is that they're not all bad, right? So bacteria, unfortunately, get a lot of bad press because we didn't used to know that they lived with us in consortia. And so the only time people noticed that bacteria were around was when they were sick. And so what I would like you to think about is that there's actually sort of two different ways to think about bacteria. So there's the traditional way, and I don't want to be a Pollyanna. There are all these bacteria that live in the environment that have no business being on a human being. And if they get in or on you, they can make you sick or they can kill you. However, there's also all these other bacteria that always do live with us, and they provide us all of these things that life wouldn't be possible without. So for example, they make lots of your vitamins, they digest your food, like if you eat plant food, the bacteria give you access to those calories, they educate your immune system to keep bad, harmful bacteria out and let these good guys in. So they do all of these things that we're just now learning about. And so that's how I would like you to think about bacteria going forward. But even if you believe that there's these two different modes for bacteria, the good guys and the bad guys, the question that I want to pose tonight is whether these bacteria are doing all these miraculous good things or whether they're making us sick and killing us. The question is, how do they do it? They're so small. How do bacteria get any bang for their buck? And that is what my lab works on. And I want to tell you how we figured out that bacteria talk to each other and they act in groups. And since this is the Discovery series, I want to actually take you on the path of where this came from, because it actually comes from a kind of funny discovery that has to do with a squid. And I'll just mention right now that Howard Hughes helped me make this video a couple years ago. So the initial observation came from people studying this animal, which is the Hawaiian bobtail squid. And this squid lives in a beautiful one-to-one symbiosis with a bioluminescent bacterium named Vibrio fischeri. So Vibrio fischeri provides the squid light, and the squid is going to use that light to stay alive. And so now I want to tell you about that symbiosis, but first I want to tell you about it from the squid's point of view. So here's the squid in an aquarium, maybe. Okay, you're going to have to help me because I have no arrow here. I have no, what? Oh, it's on the screen. Okay, then that would be a good place to look. Wait, why is this like this? Hang on, don't look at that. That's supposed to be a surprise. Don't do that. You're, all right. Okay, so the squid, is it moving? Yeah. Okay, so here's the squid. It's nocturnal. So what we've done is we've taken the sediment away and the lights are on. So the squid thinks it's daytime and it looks kind of spastic. And that's because it's never out during the day. It sleeps during the day, buried in the sand. It needs to be invisible from its predators. So what happens is if it's daylight and we give it sediment, what you can see is that it wants to escape from any predators that would see it. So it buries itself in the sand after it camouflages itself. It's thorough. You only get eaten by a predator once during the day. Okay, so now it'll retract its tentacles. And if I showed you this aquarium, you wouldn't know there's a squid there. There's actually about 10 of them in that aquarium. So this squid that's nocturnal has this really fantastic strategy to avoid predators in the day. That's to be like this. It's invisible. But of course, it can't just stay like that. At night, it has to come out to hunt. It has to come out and get food. So every night, when the sun goes down, the squid emerges, and it starts hunting around for food. And so the problem for the squid in this part of its life cycle is that it lives in very shallow water, so just sort of off the coasts of Hawaii. And since it's in just these couple feet of water, on bright nights when there's lots of starlight or moonlight, that can penetrate the depth of the water the squid lives in. So if the squid was swimming around, the starlight or moonlight would hit its back and it would cast a shadow on the sea floor, and predators could see that shadow, calculate the squid's trajectory, and eat it. So it's got this strategy for being invisible during the day, but it needs a strategy to be invisible during the night when it's hunting. And this is where the bacteria come in. So what this squid has evolved is a specialized light organ that houses this bioluminescent bacterium that I told you, Vibrio fischeri. So what we've done now is put the squid on its back, and I hope you can see these two glowing orbs that make up this light organ that house the bacteria. So what happens is living in this light organ is this single bacterium, Vibrio fischeri, but it's something like 10 to the 12th or 10 to the 13 cells are in there, and they make light. And so what happens is when the squid comes out to hunt, since these bacteria make light, that light shines down from the bottom of the squid. And what the squid has is it has detectors on its back, so it can sense how much starlight or moonlight is hitting its back, and then it opens and closes a shutter. It's just the squid's ink sac over this light organ, so the amount of light coming out of the bottom exactly matches how much light hits the squid's back. So when it swims, it doesn't make a shadow. So it actually uses, yes, biology is awesome. Yes, so it actually uses the light from the bacteria to counter-illuminate itself in this cloaking device that keeps it invisible from its predators. So now it's invisible during the day, and this is how it stays invisible during the night. But of course, the squid has this problem, and the problem is that it's got this thick culture of these bacteria in this light organ, and it can't maintain that forever. It can't keep them fresh. And so what happens is every morning when the sun comes up, the squid pumps out about 95% of the bacteria. So the bacteria are dilute, right? And then as the day goes by, the bacteria grow and double, and then at night there's lots of bacteria there to make lots of light. So it keeps it fresh, and there's only a lot of light at night. But it's actually more interesting than that, because it turns out that the bacteria don't make light except at night. And so the question is, how do the bacteria know that they're dilute in the morning and dense at night? And that, I'm going to tell you, is because they talk to each other. So now I want to talk about what's happening from the bacterium's point of view. So right in the morning, if the squid squirted out most of the Vibrio fischeri bacteria, the bacteria, this is supposed to be my cartoon of a bacteria, the bacteria would be at low cell density, right? This light organ is big, these bacteria are really small. They're at low cell density, and they don't need to make light because the squid is asleep in the sand. And so at low cell density, the bacteria don't make light. But what they do is that they make and release small molecules that are these triangles that are like hormones. And we call these autoinducers. And so what happens is that at low cell density, since the world is big, the autoinducers diffuse away, the bacteria can't detect them, and that says you're alone, and that means don't make light. But then as the day goes by, when the bacteria grow and double, since all of the bacteria are producing their share of this hormone, this autoinducer molecule, the concentration of the autoinducer increases in proportion to cell number. And when this hits a certain amount, the autoinducer concentration, the bacteria detect it. And they infer from detecting that molecule that they must have neighbors around. So in unison, all of the bacteria respond to this hormone and they turn on bioluminescence together. And this coincides with nighttime for the squid, right? And it's modulating it by squirting these bacteria in and out of this light organ. So what's happening is the bacteria don't know that it's day or night, and they don't really know that there's other cells around them. What they do is that they measure the concentration of this chemical as a proxy for cell number. And then in unison, the bacteria carry out this collective behavior that allows them to make perceivable light that in this case helps the squid, and in exchange for that, the squid feeds them and lets them grow to high cell number in that light organ. And so what was amazing about this, right, was that you could just see it. And so what scientists could do is we could just see that these bacteria made light, and we could see that they only turned on light at high cell number. So this bioluminescence made this invisible world of the bacteria visible to the scientist. And we could study it because we could actually measure light as an output of these bacteria talking to each other with this chemical language. And so what we wanted to do was to understand how does that really work in molecular level. So we brought the tools of genetics to this, and we made mutants that could or couldn't make light and asked what were the proteins and the components involved in that. And so what we found, so this is again supposed to be the Vibrio fischeri cell. So what we found was that there was an enzyme that made that autoinducer, the hormone molecule. And then that freely diffuses in and out of the cell. So the more bacteria there are, the more of that hormone molecule there is outside. And then what we found was that there was a receptor that was sitting on the cell surface, just like the receptors that sit on your cell surface. This protein sat on the cell surface, and when that hormone molecule, the autoinducer, increased to a particular amount, it could slot into that receptor, and that only happens when there's lots of cells around. And when that event happens, information comes into the cells to tell the bacteria to turn on the genes that make the enzymes that make bioluminescence. So it's a very simple circuit. The more cells there are, the more of this autoinducer molecule there is. When it reaches a critical concentration, it can find the receptor, and all the cells in unison turn on light. And so that was interesting for studying Vibrio fischeri, this sort of beautiful bacterium from the ocean. But what I think is really important about that discovery is once we sort of got onto this, we thought, well, why should this only be in one bacterium? And sure enough, in the past couple of decades, these little communication circuits have been found in hundreds and hundreds and hundreds of species of bacteria. And these are bacteria that you have heard of. They are bacteria that cause disease. And so what we now understand is that probably all bacteria have to have the capacity to understand when they're alone or understand when they're in communities so they can do different things under those two scenarios. And the way that they do it is they make these hormone-like molecules. There's always a partner receptor, and then at high cell number, these two components engage, information comes in, and the bacteria turn on all kinds of genes that underpin group behaviors. And so now we have a fancy name for this. We call this quorum sensing. The bacteria vote with these chemical votes. The vote gets counted. When there's enough to do the tasks, all of the bacteria do these tasks in unison. So it's sort of the first social biology, I guess. And then the kinds of behaviors that bacteria control with quorum sensing are always ones that it takes lots of bacteria acting together to make the task successful. So, for example, in many clinically relevant bacteria, if they don't have quorum sensing, they're not pathogens, right? So if a couple bacteria get into your body and they dribble out a few molecules of a toxin or a virulence factor, it's not going to do any good. And furthermore, it's a red flag to the immune system. So the better strategy from the bacterium's point of view is to wait and to count themselves with these little molecules to recognize when they have the right number that if all of these bacteria launch their virulence cascades together, they will be able to overcome the host immune system and produce a productive infection. So we know for now, if we make bacteria that are mutants that can't talk or can't hear, they're completely avirulent. So the kinds of behaviors that are controlled by quorum sensing are always sort of these public goods. Whenever a bacterium is going to give something away to the world, it's photons for light, it's virulence factors, some enzyme, it never gets its own back. It's only when the collective does it simultaneously that everybody profits from the work of the neighbors. So that is quorum sensing. And so once we got that far, we wondered, well, what are these molecules that these bacteria are talking with? They're not triangles, they're chemicals. And so we started to discover, we and other scientists discover what these chemical words are. So of course, the Vibrio fischeri molecule was the first one that was discovered because we've known about it the longest. So this is the actual chemical that Vibrio fischeri uses to talk with. And so then we and others started purifying these from other bacteria. And what I've done here is to just put a few different quorum sensing bacteria and the molecule that each one of these bacteria uses as its word. And I hope what you can see is that all of the molecules are related. So this left-hand part with the ring is identical for every bacteria. But these right-hand parts, these are just carbons, are a little bit different in every single species. And so you have to think of these molecules like keys to locks, which are the receptors. So each of these little tiny differences make each of these molecules slot into its own receptor and send information, but no other, just like a key in the lock of a door. So what I mean by that is if I take the Vibrio fischeri molecule and I put it on this bacterium, agrobacterium, nothing happens. And likewise, the agrobacterium, or Vibrio fischeri, is impervious to the agrobacterium molecule. So what we understand is that these slight differences in these molecules confer exquisite intra-species communication. Each of these molecules lets bacteria talk to its siblings. So these are private, secret conversations that bacteria carry out with their brothers and sisters that are for talking within your own species. And so once we got that far, we started to think, okay, these bacteria are chit-chatting. They actually have these little secret conversations where they decide to do things as a group. And we wondered, couldn't it be more complicated than that? So I told you about this bacterium, Vibrio fischeri, that lives in this pristine environment of this squid. It's the only bacterium there. But most bacteria don't get to live such a privileged life. They live in biofilms, which means they're adhered to surfaces, and they live in mixed species. So this picture, this is human skin. You have a bacterial biofilm covering your skin, right? And so this is what the skin of your elbow actually would look like under the microscope. And what you can see are all these different bacteria, and every one of these different shapes and sizes is a different species of bacteria. And so what we started to wonder is if quorum sensing is really about counting your neighbors, how do bacteria count when they're in a mixed species environment? And so we wanted to study whether quorum sensing could have a more sophisticated sort of mechanism in this kind of a scenario. And so what we decided to do was to study a cousin of Vibrio fischeri's named Vibrio harvii. So this is also a bioluminescent marine bacterium. It's related to Vibrio fischeri, but it lives free-living in the ocean. And it makes light at high cell density, but in the free-living state. And so we thought, well, this guy kind of lives in the Wild West. Maybe if we studied how this bacterium controlled quorum sensing, we could learn more about how these more wild bacteria do it. And so the reason to put this picture on here, besides the obvious cheap lab propaganda, and for the students, this is what the graduate students spend their time doing, anyway, is to just show you what we see and how we do this experiment. So the way we took this picture is we just turned the lights off in the room, and this is what we see. We took the picture from the light the bacteria are making, right? And so we're geneticists in my lab, and we don't really want to study light. We want to study quorum sensing. And so what we could do with Vibrio harvii, and we did this with Vibrio fischeri, is we can make mutants. And we can just plate them out on these Petri plates and turn the lights off in the room and look for bacteria that are making light when they shouldn't be or not making light when they should be. And that's how we found it's super easy, right? That is how, and then we pick those guys that are misregulated and we find out what's wrong with them, right? So it's a really easy experiment. You just have to be able to turn the lights off in the room and find the dark ones and the bright ones on these Petri plates. Okay, so by doing that experiment, we found the components of the Vibrio harvii quorum sensing system. And so similar to what I told you before, and not surprisingly to us, we found an enzyme that made an autoinducer, and I actually showed it to you a couple slides ago, and it had its partner receptor. But when we eliminated these genes, this bacterium still had quorum sensing. And so we went hunting again, and what we found was that there was a second quorum sensing system. So we found a second enzyme that made another molecule. It had its partner receptor, and then all of this information comes into the cells to tell the cells to turn on and off light. And so we wondered, well, why did evolution put this second system here? If these two molecules don't encode different pieces of information, having two systems isn't better than one. And so what we did was we cloned all the genes for this system. And you know how we have the human genome? There are thousands of bacterial genomes. So we could take the genes, the sequences of these genes, put them into the database and just ask, does any other bacterium have them? And what we found was that every bacterium that was in the database had the gene to make this molecule. And so we named that gene Lux S for the synthase and Lux for the god of light. And what we found was that all bacteria at that point had this enzyme, and they all seemed to make this molecule. And so what we did then was we purified that molecule and asked, what is it? And I'll call this autoinducer 2. And what we found was that no matter what bacterium we purified the molecule from, the molecule was identical. It was these 5 carbons decorated with these oxygens. And so the point that I want to make is not what the structure is. The point I want to make is this is very different from what I told you about the other system where each species of bacteria makes a slightly different molecule. In this case, every species makes the identical molecule. So what we think that means is that this is a sort of interspecies communication language. This is like the bacterial Esperanto, the common or trade language that bacteria have. And so what we showed then is that bacteria can talk across species and influence one another's behavior because they all use this same language, which we call this autoinducer 2. And so now what we think is that at a minimum, and I'm not done yet, right? I mean, I still have a job tomorrow, I hope. Anyway, but what we think now is that bacteria have to at least be bilingual, right? They're multilingual. They have to have some mechanism to count their siblings, and then they use autoinducer 2 to say other. And so the computation that we really think bacteria do is the following. We think the first thing they do is they just sort of scan the environment with these receptors, and they ask, are there any molecules there? And that begins to tell them whether they're alone or whether they're in a community. And then they start to turn on and off group behavior genes. And then the more sophisticated computation that we showed they do is that they actually measure the ratio of these molecules. And then they change what genes they turn on and off based on whether they see a lot of this one, which means me and my kin are in the majority, or they see a lot of this molecule, which means me and my kin are in the minority. Right, so now what we think is that the quorum sensing molecules allow the bacteria to count, but encoded in these molecules is something about how closely or distantly related I am to you. And the bacteria tailor their activities based on whether they and their family are winning and losing in these different mixtures that actually exist in the wild. And so there's going to be more molecules than this, right? There's probably more languages, but this is the basis, we think, of quorum sensing. And so now we know that this happens. We know these bacteria have these group behaviors. And I've already told you that quorum sensing, and I'll tell you right now, both of these molecules control biofilm formation, which is how these bacteria sit on surfaces. That's the first step in the infection. They have to adhere, and then so they do that as a group. And then as a group, they have to release their poisons, their toxins, to make us or animals or plants sick. And so now the sort of practical part of this is, could you actually interfere with quorum sensing to make new kinds of medicines, right? And so there's sort of 2 strategies that one could think about. The first one would be if you wanted to interfere with this intra-species specific language. So lots of times, if you're sick, the physician knows what's wrong with you, and you want to be treated with a very narrow spectrum antibiotic that treats that bacterium. So maybe you'd like to make an anti-quorum sensing molecule that actually is very species specific, like a narrow spectrum anti-quorum sensing therapy. The other strategy is the opposite one. Sometimes if you're sick, your physician doesn't know what's wrong with you. And so typically, initially, you're treated with a broad spectrum antibiotic to try to cover all kinds of possible pathogens. So in that case, maybe what you'd like to do is to target this interspecies communication system in order to make a broad spectrum anti-quorum anti-quorum sensing therapy. And so we're trying to do both of those things, and I'm only going to explain the logic for one of these systems because the steps are exactly the same, right? So you can use your imagination for the second one. But before I tell you about our steps into that, I want to take a little interlude and actually talk about why might you want to do this, right? Like, why would you want to make an anti-quorum sensing therapy? And I think that the obvious reason is because we have this global problem with antibiotic resistance. And I know you guys hear about this and read about this all the time in the newspapers, so I thought I would take just a couple minutes to talk about what antibiotics do and then why bacteria or how bacteria get resistant and why it's a problem. So the first thing is, what are antibiotics? And so I just put a few famous ones on this slide. They are chemicals. They're simply chemicals, and these chemicals kill bacteria. They are poisonous to bacterial cells. Okay, so those are a couple antibiotics that you've probably heard of. And so then the question is, so why are these molecules poisons, right? Not all molecules are poisons. And it's because antibiotics target different essential activities that bacteria do. So some antibiotics, for example, bind to and shut down DNA replication. So if bacteria can't make another copy of their DNA, they can't divide and make two daughter cells. So the bacteria get killed by an antibiotic because they can't make more DNA. Some antibiotics that you've taken actually interfere with the ability, so the bacteria can make their DNA fine, but they can't translate it into these other two important biomolecules, RNA or protein. And so other antibiotics shut down these other functions. And so if a bacteria can't make any proteins, it also can't divide. Some antibiotics simply make the cell membrane real flimsy. That's like penicillin, and so the bacteria pop. So those are sort of the key ways that antibiotics work. They shut down either these essential functions or they make the bacteria weak, right? And so the bacteria can't divide. And so what happens is every time a bacterial population is growing, random mutations happen. And occasionally, those mutations will happen in these components of the cells. So the way bacteria get resistant is if fortuitously they get a mutation that disables the antibiotic from doing these activities. So for example, bacteria have pumps to pump out wastes, and if they could get a mutation that in addition to waste, they pump out the antibiotic goes in, the antibiotic goes out. It's not going to kill the bacteria. So that's resistance. They could get a mutation in some of the machinery that's for making proteins, and now the antibiotic simply can't bind. That's resistance, right? They could actually have an enzyme that's supposed to do one job, but to degrade things, but it degrades the antibiotic. So the antibiotic goes in, but it disappears because this enzyme degrades it. And the other kind of mutations they can get is they can just get mutations in the components of the membrane that make them tougher. And so these antibiotics don't make them flimsy, right? And so it's not magic, right? Bacteria divide, mistakes get made in their DNA every time they divide, and if they happen to get a mistake in some of these components, they and all of their offspring will be resistant to the antibiotic. And so what happens is when we give antibiotics, like if a person or animal or they're in the environment, if you have all these bacteria that are going to get treated, these are just supposed to be bacterial cells, they're going to be treated by an antibiotic. And some member of that community fortuitously gets a mutation that makes it resistant. What happens is when the antibiotic is applied, so that's supposed to be my blue squares, all of the bacteria that don't have that mutation, they die. So the only bacteria so usually this bacterium would be in a minority, but since all of these competitors and all these other bacteria die, then the only bacteria that can grow are the resistant mutants and their offspring. And this is why resistance spreads, is because an antibiotic kills off all what I'll call the wild type cells, and so it gives a tremendous growth advantage to any mutant that happen to acquire resistance. So that is the global problem that we have with antibiotic resistance, that you guys know about. And so then the question is, well, could you think about this a different way? So what I've just shown you is that with a traditional antibiotic, most bacteria get killed, and so a resistant mutant that might have been in the minority can grow and take over. If you could make an anti-quorum sensing therapy, so here's the logic. We haven't done this yet, right? Now what you're doing is you're making bacteria that can't talk or can't hear. You're not changing their growth. You're telling them don't express these quorum sensing behaviors. So in that case, if we could apply this anti-quorum sensing therapy, this guy, you would get bacteria that are resistant to the anti-quorum sensing therapy. But all of its neighbors don't get killed. You just shut off quorum sensing. You change behaviors. So in this case, this guy stays, presumably, as a minority, right? And these guys can't do quorum sensing, so virulence doesn't happen. This mutant would be like, come on, you guys, let's do quorum sensing, but nobody else participates. So the behavior wouldn't happen, and the fantasy is that you wouldn't get this immediate selection for growth of the resistant mutant. So that's the logic behind trying to make what I'll call a behavior modification therapy instead of a life or death therapy, which is what a traditional antibiotic does. Okay, so this is what we're trying to do in the lab right now. And so now I'll just finish by going back to this quorum sensing story and tell you about the steps that we've made so far. So what we decided to do is to try our test of this idea with a bacterium called Pseudomonas aeruginosa. And this is a notorious pathogen. So you guys have heard of the disease called cystic fibrosis? So people who have that disease have a genetic mutation in their lungs. So they can't clear their lungs. Like you and I breathe in all kinds of gunk, and we have ways to keep our lungs sterile. People who have cystic fibrosis can't do that. So they always have this polymicrobial infection. But what happens is typically when a person is in his or her teens, for reasons that we don't understand, they become permanently colonized by Pseudomonas aeruginosa, and that's what kills people that have CF. They die of a Pseudomonas aeruginosa infection, and that is because Pseudomonas has quorum sensing. It gets into the lung, quorum sensing launches, it makes this biofilm that makes it stick to the lung, and then in unison, they secrete all of these virulence factors. They're just trying to get nutrients, but the consequence of all of these virulence factors is this lung damage that kills people who have cystic fibrosis, and it is a terrible, devastating disease. And we know that this pathogen is involved in mortality, and we know a lot about its quorum sensing system. The other thing about Pseudomonas aeruginosa is it's really sticky. So you know how it's becoming like the 19th century again? You know, you go to the hospital to get your gallbladder out and you come out with the bacterial infection again? That is because of this bacterium. It loves to form biofilms. So it coats surfaces, catheters, stents. It's in hospitals, and it's a real problem because it's super sticky, so it's hard to get rid of, and so it causes these hospital-acquired infections that aren't actually the thing the person went in to get cured for in the first place. So it is a terrible emerging pathogen for two reasons. One, the cystic fibrosis, it kills those people, and the second is because it's kind of running all around hospitals. So both biofilms and virulence are controlled by quorum sensing in this pathogen. So this is sort of the emerging pathogen that doctors are very worried about in this nation. So we thought, put your money where your mouth is and go after this guy. Okay, and so just to reiterate, what I told you is what we already know is that if Pseudomonas doesn't have quorum sensing, it will not make a biofilm, and it will not express these virulence or toxic factors. Okay, so we know that. So we wanted to see, could we interfere with its quorum sensing system? And so we went after this species-specific molecule, right? And so then to remind you, this is the molecule, the autoinducer that Pseudomonas talks with. It actually makes two of them. One has a 4-carbon tail and one has a 12-carbon tail. So what we did, so that's the natural molecule, is using synthetic chemistry, we made a molecule that doesn't exist in nature that looks kind of like this molecule. So if you look at this part of it, except for this sulfur, there's an oxygen here. So this part of the molecule looks like the autoinducer, but then we put a big bump on the end. Right? And so what happens is that is an inhibitor of quorum sensing. So if the molecule on the right slots into the receptor, quorum sensing happens. If our molecule, the synthetic one, slots into the receptor, it gets stuck there. Quorum sensing doesn't happen, but this molecule can't get in either. So it sort of blocks the receptor from allowing the real signal to get in. So this is a quorum sensing antagonist, an inhibitor. Okay, so that's our best molecule, and we know how it works. Okay, so that's the logic. So now I'm going to try to prove to you that maybe there's merit to this idea of thinking about this as a new kind of therapy. So remember I told you for Pseudomonas to be a pathogen, it has to make a biofilm, and then it has to secrete all these virulence factors. So we said, what happens when we supply that molecule? So this is a Petri plate with Pseudomonas on it, and it's making a biofilm. So what we do is we simply put a little drop of Pseudomonas cells in the middle of the plate. And then if we wait overnight, they spread out into this beautiful but bad pattern, right? And they make a biofilm, and they grow, and they spread out from the center, and they cover the plate. So that's a Pseudomonas biofilm. If we put our molecule into that Petri plate, and we put the Pseudomonas in the middle, they can't make the biofilm. So that was really good news for us. That's step one. We can shut down biofilm formation by blocking quorum sensing, not growth, not changing growth. Okay, so that's step one. Step two is that Pseudomonas launches releases all these virulence factors. So we wanted to test whether our molecule could work there. So Pseudomonas, I keep drawing green because it makes a virulence factor. So the protein that kills, or the molecule that kills lung cells is called piocyanin, cyan. So it's a molecule that's a killer molecule, but it actually has a green tint to it. So if you just grow a high cell density quorum sensing culture of Pseudomonas, the whole culture turns green because they release this piocyanin virulence factor, and that just turns them green. So if we make a quorum sensing mutant, right, can't talk, can't hear, what you can see is that the culture is colorless in this test tube. So that shows you that quorum sensing regulates production of this green virulence factor. If we take this wild type bacteria, but now we add that inhibitor, the quorum sensing inhibitor, what you can see is that the culture is very pale green. It turns out that we can shut down 90% of the production of this virulence factor. So those two experiments made us pretty confident that this might be a reasonable strategy, but of course it doesn't really happen in a test tube. So what we wanted to do once we got those results is to try, could we shut down quorum sensing and stop an infection in an animal? And so here's the last experiment I'll show you tonight. So what we do is we have an animal model for cystic fibrosis. And so we look at how many animals are living over time. And if we don't give Pseudomonas, obviously, the animals are perfectly healthy, right? And so they live a lot longer than 30 hours. Okay, if we infect the animals with Pseudomonas, what you can see is that all of the animals die in a day. I mean, that's why we use this model system. So if we do that experiment, but simultaneously we give our anti-quorum sensing molecule, what you can see is that we can actually cure the animals. And so now we're pretty confident that this might be an interesting way to think about, they're not really antibiotics, but think about antimicrobials, right? And so we're moving forward with this. And so I want to be really careful here. This is not a medicine. You'd have to take a pill this big and it would kill you, because we haven't done any of the chemistry to make this safe and really potent yet. Those are the next steps in the lab. But this is our newest experiment that makes us think that we should actually keep trying to pursue this idea. So the things that are on our horizon right now is to make the molecule medicine-like. And what I mean by that is that medicinal chemists take what I would call lead molecules, and they build in the features that make them stable in your body, make them not harmful, that make them not immediately go to your kidneys and get excreted. So those things have not been done to this molecule, but those are the kinds of things we're working on. And then remember, Pseudomonas is this terrible biofilm former. So what we'd also like to do is we're working with engineers now to try to dangle these molecules off of surfaces or off of catheters. Like maybe you could make infection-resistant, stents or catheters or hospital machines if you could actually coat them with anti-quorum sensing molecules. So we're trying to put our inhibitors on those as well. And then, of course, I'm just telling you this Pseudomonas story, and so we work on lots of different pathogens with similar logic in the lab. And then the last thing, the next thing is that business I told you where we hope that an anti-quorum sensing molecule will take longer before the bacteria get resistant. We have not proved any of that yet, but once we have these inhibitors, we can check how long does it take for the bacteria to develop resistance. They will get resistant. The only hope is that we will get a longer shelf life out of these so while one of these younger people thinks up the next strategy, right, than we do for traditional antibiotics. And so then to finish, here's the big summary. What I hope you think after this talk is that bacteria can talk to each other. They don't have voices, and so the language they use is chemical. What I hope you also think is that bacteria are multicellular. They have these collective behaviors. And so what we hope in our lab is that by trying to understand how group behaviors work in bacteria, which are simple systems, we'll actually learn the principles for how collective behaviors happen in higher organisms. Bacteria were the first organisms on Earth. We all evolved from bacteria, and so we believe that the principles for how you get collective behaviors to happen robustly are going to be embedded in these bacteria, and that if we study them, we can help our colleagues that actually study higher organisms to understand more sophisticated systems. What I showed you tonight with those two molecules is that bacteria can distinguish self from other by using different chemicals. I would argue that's what happens in your body. It's not like your heart cells get all mixed up with your lung cells every day, and that's because different chemistries tell these different groups of cells who they are and what their jobs are. And again, we think bacteria evolved that kind of sophisticated trait long, long ago. And then, of course, there's these practical parts, which is we can develop strategies. I told you about impeding quorum sensing, but to go back to my second slide, remember, most of the bacteria that we encounter are keeping us alive. And so there's a totally other part of this strategy, which is that maybe we could make quorum sensing better. We could make pro-quorum sensing molecules that we could use for agriculture or health or industry or making biofuels. So if we could actually improve quorum sensing, we could get good bacteria to do better things. And that may actually be the way forward in medicine as well. It could be that we should just stop fighting against harmful bacteria and get these good bacteria to do the work for us. And so lots of people are working in that area as well. And then the last thing I'll do is I'll finish with a confession that once we sort of got onto these ideas, we thought, if we can think of these bacteria probably thought of this billions of years ago. And so sure enough, we and lots of other scientists have sort of gone out into the environment, where these bacteria are fighting in fierce competitors. And sure enough, they already make quorum sensing inhibitors. They make enzymes that cut the rings off these chains. They eat each other's autoinducers. They free ride, they cheat, they eavesdrop. And those things that these bacteria have done, the idea that Bonnie Bassler and some 23-year-old chemistry graduate student are going to do a better is ridiculous. What we should do is go out there and figure out what these bacteria evolved, because the strategies that exist in the natural world have been tested over billions of years. So we are bringing those into the lab and then tinkering with those. But those are going to be our starting blocks, our starting steps going forward. So that's sort of where we are right now. And then I want to finish by saying that I get the very good fortune to get to come give the talk. But the people in this slide, this is my gang at Princeton. And every idea that I told you, every experiment that I told you was thought up by somebody's brain, not that one, in this group, and was done by their hands. And their belief, this is to the young people, that actually doing curiosity-driven discovery science can actually change the way people think about the world, and that our lives really matter to do these kinds of things, even if you're working on a glow-in-the-dark bacteria, right? That if you're curious and you think you're working on a fundamental problem, in fact, if you work it hard enough, these amazing things can come out of it. And so I just want to say, especially for the students that are here, that if you're interested in science, doesn't have to be biology, but science, you should just stick with it, because it's so much fun. You get to be the discoverers. You get to think about things that nobody's ever thought about before. And you always get to work with really fun, smart, curious, nice people, right, that want to understand how the world works. And so it's a real privilege to be a scientist. But for the young people, what I am telling you is the people that make discoveries are the people that are in their 20s or 30s. People like me go around talking and typing and hanging with them. People like you make the discoveries. And so if you like that, you should stick with it, because it's you guys that change the way we understand how the world works. And so anyway, I want to thank you guys for listening to what we're doing down the road, up the road, I guess, in Princeton, New Jersey. I thank you for coming out in the rain. I thank Jerry for having me. And I think we take questions? Yes, okay. Thank you. Yes. Is that a question? You just did.
Sorry. If bacteria can talk to themselves, when they mutate, if the other bacteria doesn't want the mutation, can they reject it?
They cannot reject it. So if a bacterium gets a mutation in its DNA, the same as we do, it will pass it down to all of its daughters. Right? So you don't get to choose that. So you are defective once that happens. That's a great question. Oh, yeah.
So you were talking about how bacteria like have their own quorum impeding things, and that it's been doing it for like billions of years. So why do you also think that they're going to have resistance if bacteria have been doing it themselves for billions of years and they can keep on doing it?
Right, agreed. A resistance it's a fortuitous mutation, right? So I think out in the wild, a bacterium, this is kind of your question, if it gets resistant, like if it is incapable of doing quorum sensing, it gets outcompeted out in the wild, right? Because being able to participate in collective behaviors gives you an advantage. I think in medicine, right? If you had an anti-quorum sensing molecule, right? Even if a bacterium got resistant, it wouldn't get an advantage in my scenario. But I believe they're getting mutations all the time that make them resistant to quorum sensing, but they get outcompeted because they can't, because then they've lost actually what I think is a very valuable trait, which is to be able to participate and to talk and be able to count. Yeah, that's a great question. And they will get resistant, yeah. Okay, over there?
Yes. So first of all, wow, wow, wow. This was so cool.
Oh, you are silver-tongued
So,
devil.
but I have two quick questions. The first one is, how do you find these anti-quorum sensing molecules? And the second one is, you said these are kind of like hormones. Is there any relationship in the evolutionary development of hormones?
Okay, so I'll answer the second one first. So maybe I meant they're like hormones in the sense that they're sensory information. They are not the evolutionary precursors to hormones. What I meant is that your cells respond to molecules around them. We call them hormones. These bacteria respond to molecules that we call autoinducers, but they are analogous to hormones. But they did not come one from the other. Sorry if I confused people about that. The second question, how do you find the anti-quorum sensing molecules, right? So that's actually fun. So it turns out that chemists make molecules, right? And they make them to learn the principles of chemistry. And what all the chemists with the NIH have done, or lots of chemists done, is that they make them to make new reactions, to make new shapes, to sort of probe how chemistry works. The same way a biologist thinks about biology. And the chemists have all gotten together because they've made all these molecules. And for them, in many cases, the making is the thing. And they've actually put them all into a repository, right? And then they're sort of in like arrays, one molecule in each little space, and then they've donated them so that the biologists can come and actually use their assays and ask, does any molecule that somebody made for completely other reason hit my receptor? And so we simply, with a robot, you can actually screen through hundreds of thousands of these molecules that have all been put into this repository. And then you see after the fact why that molecule might be inhibitor. It kind of looks like the real autoinducer, right? And that's where we got the original ones. And then we do chemistry. We start with those, and then we do chemistry to make them more potent, more selective, less toxic to human cells. Yeah, but it's a really nice thing where all these biologists and chemists, so you can look for your anti-cancer drug, you can look for your anti-Alzheimer drug, right? And we looked for our anti-quorum sensing drug. And so everybody's screening these. It's a wonderful resource. Yes?
In human tumors, metastasis sometimes occurs when there is a critical mass of tumor cells. Do human tumors have some kind of system which tells that it's time for the tumor to private?
Right. Did you guys hear that? Sometimes when you have tumors, cells escape. That's called metastasis and goes somewhere else. And it looks, and so far, so I'm not a cancer expert, right, about which of these cells decide to leave and go to the next place. But so far, there isn't quorum sensing in higher organisms yet, right? So the answer is I don't know, but I don't want to overstate that. There's no evidence. I am brainwashed. Of course I think there's going to be quorum sensing in higher organisms, right? But no quorum sensing molecule has been found yet, but we are looking, right? And so I think the place to look, actually, what we're doing is in yeast. So yeast are microbes, but they're more like human cells than they are like bacterial cells. So they're sort of simple higher organisms. And so you don't go all the way from bacterium to a human cell. You kind of bootstrap your way up there. And so lots of people are looking for quorum sensing in yeast. Who am I? Am I supposed to be, taking orders?
So my question would be, can they read our hormones as if they were quorums?
Right.
And if so, can we discriminate against them to kill
Right.
those ones?
Right. So that is actually, right? So now you guys know we have this microbiome with these 600 or 1,000 species, right? It's a chemical treasure trove in there, right? And so the real question is, and we talked about this in the class, is there interkingdom communication? And it should matter whether these are beneficial or hostile relationships. So the first hints are coming out that at least that there's eavesdropping, right? And that these bacteria are driving some of what your human cells do. And likewise, your human cells might be making molecules that the bacteria are responding to. So mimics, quorum sensing mimics. And so there's starting to be evidence. I think that would be awesome, right? Intraspecies, interspecies, interkingdom. I think that, these bacteria that like live in your, they're not willy-nilly. There's geography, there's the right numbers, they're always, organized, in the same way, right? There's got to be an incredible, I think, dialogue, not a monologue, going on in there. And lots of people are hunting for those kinds of things now. I think that's going to be the next big deal in the quorum sensing field. Mm-hmm.
Since you're targeting the second system that exists in all bacteria, how would you be able to distinguish between the good and the bad?
Right, you guys get that? Right, how do you tell the difference between the good and bad? So the molecule that I showed is it was for the first system. It's an anti-Pseudomonas molecule. And we screened against other bacteria that it wouldn't hit other bacteria. So that molecule that I showed you is quite specific. But you're still right, because I did say we want to do this interspecies thing. Right, and so everybody get that? The molecule I showed you is for Pseudomonas. It goes against the intraspecies communication arm of the circuit. If you could make this fantasy anti-universal autoinducer molecule, your question is, but all these good bacteria make that molecule too. How do you distinguish? And my answer is you don't. So remember, when you take a traditional, and now here we're banking on the 80 years that we've used traditional antibiotics. When you take a traditional antibiotic, penicillin, tetracycline, it also doesn't discriminate, right? And so sometimes you get a stomach ache, women get a yeast infection because it kills lots of different bacteria. The miracle of this microbiome that's in you is that there's these cells that are called persister cells that aren't affected by the antibiotic. We don't understand this, but you have these little pockets of cells that after the antibiotic gets taken off, they repopulate. The question now is, how could antibiotics have ever worked? And they do. They are the greatest public health measure, that and clean water, right, ever in humankind, right? So they really work. And what's awesome is that your body repopulates with these little pockets of persister cells that somehow aren't killed. And so now if we could make an autoinducer 2 molecule, right? In our case, you're right, everybody would be like, they wouldn't be communicating right? And that would give the immune system time to mop up the bad guys, which is what it's supposed to do. And then when you took the therapy off, this is all fantasy, in that case, you haven't actually killed everybody, right? Then they would go back to, we have not proved that, but we know a lot from the use of real antibiotics, right? So that's the logic. Mm-hmm.
I was wondering, the Bacterial Fishery, the
Mm-hmm.
relative of that.
Mm-hmm. Harvey I.
Yeah. So the Bacterial Fishery produce bioluminescence, but it's at very high concentrations. Is that the same case as that, or is it just actually respond to the lights being in the dark?
So the Vibrio fischeri is a funny guy. So it does what Vibrio harveyi does, but there's a cue. It doesn't make it free living. So the squid must be somehow also controlling it. If you take the components out and you put them in E. coli, they all work. But somehow the squid is also controlling Vibrio fischeri, that it only turns on light inside the host. Harveyi turns it on outside.
And does it only turn it on when it's in high concentrations, or
Yes.
responding to the light?
No. Sorry. They don't perceive their own light. It's only perceiving that molecule. So we can trick them. You can take dilute cells, and we can make all these molecules, right? And so if you add the molecule, the autoinducer, to dilute cells, they will turn on light because they don't know. They're just measuring that chemical, and you can trick them into thinking there's lots of cells around just by adding the autoinducer.
When you're turning off the light, you're just seeing the light.
We are just being able to, we are turning off the lights. Yes. So I get you can't see that color of light against this light. So we turn off the light so we can see it. They don't understand that.
So I'm not
Lucky
going to
for
turn
us.
off the
I have a
lights, but I
question.
am going to turn off the
I'm
questions.
going to turn off the microphone.
One more question, please.
But thank you very much, Brian.
Thanks. Thank you.
Hey, Karen, how are the tornadoes? Can they go home? Yeah, sure. I think the tornado warning is done, so.